I'm the host, the token host for today. My name is Dr. Jenny Carr. I'm in the Department of Biology, and I'm also the chair of the John S. Toll Science and Mathematics Fellows Program. It's a mouthful. And one of the things that this program offers is funding for these collaborative projects. So two of these students have done their research projects in summer 2020 remotely, thanks to COVID, but we're still able to do some research. And then two of our presenters are currently doing research in the lab and or field with faculty. So I'm really happy that we could pull the four of them together. There's great diversity in different departments. So I think we'll all learn a lot. So I've asked them each to keep their talks to about 10 minutes each. And then after each talk, we'll pause for a question or two and then move on to the next talk. And then afterwards, we're going to leave it open for sort of a mix and mingle Q&A session for our Toll apprentices and fellows. So if the apprentices and fellows have been able to join us today, welcome. And I hope that you'll take advantage of tapping into some of your peers' experiences and getting to learn about the program, what research is like, and all the cool experiences they've gotten to have. So with that, I'm going to introduce, well, I'm just going to say that Evan will be presenting first. And I'm going to ask Evan to introduce himself. So what's your name, major, what year you were in school, and who you worked with, and then you can just start into your research project.
Thank you so much. Hi everyone, I'm Evan McCarthy. I'm a junior at Washington College and I'm a math computer science double major. My summer project was under the advising of Professor Shaun Ramsey of the Computer Science and Math Department, and I think we've got a really cool thing to show you today. So I'm going to open up a screen share and can you guys all see it? Everything good?
Looks
Awesome.
good.
Awesome. So the project that I worked on with Professor Ramsey this summer is a project on optimizing the LYAPUNOV calculation. And the LYAPUNOV calculation is a measure of chaos in a system. Now, chaos is a mathematical measure or a mathematical description of systems that have large changes in their output for small changes in their input. And a really great example of that in the real world is weather forecasting. In a weather model, a small change in temperature or a wind gust here and there can drastically affect the results for the forecast later in the week. That's why the weatherman or woman always gets the weather forecast wrong when it's super far out, because all it takes is little shifts in the initial conditions to make large changes in that further out forecast. Now, the Lyapunov exponent is usually calculated using a slow iterative method which is computationally intense and doesn't scale to large amounts of computer power very well. And so what our research is testing and comparing a new method for calculating this measure of chaos called the linear approximation method. And what we're testing that method on is something called the logistic equation. And so this is a recursive equation, meaning it maps onto itself. So the results of one iteration will then affect the results of the next iteration. And it's a really great example of chaos in the real world and has real world modeling applications like the population of rabbits over time. And it's not too complex for us, so we can use our normal computing hardware to iterate and calculate over this. And so the big deliverable of this project was a graphical user interface, a program which you can run on your computer, which will in real time compare the old method and the new method. And I coded this one up in a computer programming language called Python. Now, Python is a really versatile and modern computer science language with vast applications in data science and data modeling. And I decided to use a library, which is a package of code that a group of people out in the world has worked on to make projects like this easier. So I don't have to, from scratch, develop the graphical user interface and the plotting software. We could do the computations and then hand that over to the library so it can do the heavy lifting of putting it up on the screen. And Tkinter is that library. And that allows us to, in real time, change parameters or use different points or different equations, and it plots all that with a library called matplotlib. And it's this really cool, powerful library which can draw all kinds of plots, from simple shapes and y equals x plots like in middle school to huge complex operations and even 3D plots. But we didn't get into that. We don't have to do that. The final program that we developed was over 800 lines of code, and using Tkinter and matplotlib, we got some really cool results. Now, Tkinter is a very powerful program, but very, very quirky, and I learned a lot along the way about computer programming, graphics visualizations, and the more crazy architecture components of Python. And it's because it likes its things its way or the highway. And so if you were trying to bend and move it around to do some things that you wanted to do, it was very resilient. And there was many times when me and Dr. Ramsey were digging through the source code and not so great documentation to try to find out how to do things. And we had to use some really clever tricks in the code to get certain things to work, like the plots refreshing in real time. Now, matplotlib, like I said, was the program that we used to actually draw the plots of data, and it is very well documented, and there's all kinds of different plots that we can use. At the end of the presentation, I'll show some of the different iterations and kind of exploratory programs we did, and there's some really cool plots there. So now we get to the actual program, the deliverable of the research, and this is divided into two main parts. Let's see if I can get... On the left side here, we've got the actual plot and the results. So this plot will show in real time all the parameters or equations that you've passed into the program, and then at the bottom, the two comparisons. So we've got the old method, the iterative LYAPUNOV exponent, and the new method, the LYAPUNOV exponent. And the old method is our slower iterative one, which takes a while to compute, and then the one on the left is our newer one. And so not only can, with this program, we compare the results and their accuracy to see if there's any really big discrepancies, but the time it takes to compute them, which is one really cool aspect of this, that we have a 6 times reduction in the amount of time it takes to compute this. Now, on the right side of the program, we've got all kinds of parameters. On the top, you could enter in your own custom programs in the point entering mode, and then change some of the parameters of the LYAPUNOV exponent. We've also got some parameters for a font size, to make it easier to see, and a slider for precision. So we don't always need all 10 digits of precision that this program can use. It makes computing time slower and heats your computer up a little bit. So when we don't need that, we could slide down our precision and still get really accurate numbers, but not to the crazy accuracy that you might need if you were calculating to 10 decimal places. Imagine doing that by hand over and over again, dozens of times per second. Doesn't sound too fun. And then the other component of this is the logistic equation plot. So the previous one is where you could enter in your own equations manually point by point, or using points selected from the logistic equation. There's an override toggle right here, which allows you to toggle on this mode, and then you can select the number of points that you want to take from the logistic equation. So right here we've got an example of 3 points sampled from the logistic equation, shown by these colorful lines here. And then this whole blob here is actually what's called a cobweb plot. And it's part of this chaotic logistic equation business, and it helps us determine chaos. And not only is it a great visualization to show how the program is arriving at some of these results, but it looks really cool, and you can find some really cool plots that just make it go really, really crazy. These are some other examples of plots that we've generated. There's, of course, an enormous range you can make, but these are some of the cool ones. This is a higher resolution version of the default one, and then this is a different sampling from the logistic equation. So right here we've got 1, 2, 3, 4, 5 points sampled, evenly spaced along that horizontal axis, and then a different example of a cobweb plot. So this one isn't as dense, but it's still arriving at that nice center there. Now, I promised you some really cool plots, and I think these deliver. So in order to learn about chaos and practice with the different GUI and plotting aspects of this, Dr. Ramsey had me go through a couple different types of plots and generations. So we've got a kind of a beta version of the graphical user interface, where I was practicing the input, and then we've got a bifurcation plot, which is a really, really cool component of that logistic equation, and it makes all these really cool colors that line over itself. It's kind of like a piece of art for nerdy people like me. And then other plots over time, and then superfractals, which is a component of Dr. Ramsey's research, and then other iterations and cobweb plots that we went through the system. Now, we had a really crazy cool program come out of this, but there are still some limitations. Tkinter gave us a lot of trouble with it being finicky and didn't really adapt to some of the things we'd like to do. So in the future, we might have chosen a different plotting or a different graphical user interface library, and there's lots out there to explore. Python is also notoriously limited to one computer brain. So our modern computers can have 2, 3, 4, 6, even in the server ones, 64 of their individual brains, and Python can only ever take advantage of one at a time. So in the future, we might be able to get around some of those limitations with more advanced libraries, or even a different language than Python, so the program can run even snappier and even faster. And that's the presentation. Here's the works cited. Thanks, guys.
Thank you. Excellent. Lots of air claps you're getting.
Yeah, no one wants to have their microphones on. Trust me.
No.
And now I'll open it up to any questions, or if anybody wanted to see a slide of the presentation again, please let me know.
And if you'd rather type your question in the chat, go ahead and I'm happy to moderate.
If you don't want to say it out loud, please do. And we'll also, like was said earlier, be staying behind a little bit if you want to ask a one-on-one question there. All right. Thank you, guys.
Evan. This is Suyog. Very nice presentation. I have a very quick question about one of your plots, the 10 plots.
Absolutely.
Just wanted to understand if you can go to one of these slides. You have different color coding for the lines.
I'm sorry?
Is there any meaning behind the different colors for the lines? So for example,
you have the option to change your map colors to different things along here. The different color of lines is actually a default part of the matplotlib plotting library that I decided to leave, not only because I thought it looked really cool, but it added some color to these otherwise really monochrome graphs. So it's easy to see. For example, over here, you can really see how those are separate lines. And when you get up into 20, 30 different points selected, the lines, you can actually kind of see it close to the curvature of the actual logistic equation a little bit better. So it's mostly an aesthetic thing. They don't actually have a mathematical function to them.
Excellent.
There's a question when you could start predicting weather with the program. Well, maybe instead of predicting weather with this program, this will make the predictions faster and better. So maybe we might be able to get a little bit better, maybe an hour or two better out, but I'm still never going to trust the forecast.
Hey, an hour or two out could save some lives, so it could have really big implications.
Definitely. Thank you so much, guys.
Thank you. Excellent job. And you kept it at a great time. I didn't have to cut you off for going too long.
Glad to hear it.
It was great. Okay, next up will be Peyton. So I'll let him introduce himself, his advisor, what year you are, and what's your major.
All right. Hi, everybody. I'm Peyton. You said, oh, what year I am? I'm a senior. My advisor was Dr. Suyog and Dr. Tex in the physics department. And yeah, was that it, or was there something else? Was there something else?
Nope, you did it. That's great.
Okay, cool. All right, let me pull this up here. All right, can everybody see that?
Looks good.
That's good. All right, cool. Let me go into present here. All right. Awesome. Let me get this out of the way. Okay, so my research project was background modeling for the dye-Higgs search with the Atlas detector at CERN's Large Hadron Collider. Let me start it. Yeah. Okay, there we go. So here's the talking points I'll be going over. So we have our introduction, the methods that we used to do this research, some of the results and observations, a conclusion, and some acknowledgements. So here's the introduction. So the fundamental building blocks of our universe are elementary particles, which are called quarks and leptons. And these particles, you can see that these are over here on the right, or on the left side, there's quarks there. So they make up protons and neutrons, which in turn make up electrons. And you can see how it just kind of builds upon itself until you eventually get to matter, the stuff that, you know, we are made of and everything's made of. So these particles are mediated by force-carrying particles called gauge bosons. And all of these quarks and leptons and gauge bosons are a part of a larger theoretical structure called the Standard Model. And in particular, the particle we're interested in our personal experiment or our research is the Higgs boson. So a little bit about the Large Hadron Collider. This is what I was using data from here, working with my research, and yeah, we were that's how it was working. So the Large Hadron Collider is the world's largest particle collider. It spans 27 kilometers in circumference, and its function is to accelerate protons near light speeds in an ultra high vacuum tube towards each other until they collide. I don't know if you can see my mouse, but you see those two yellow arrows that say P and P. Those are the protons, and you smash them until they have a head-on collision. When they undergo a collision, they create a shower of particles, which can be detected by some of the detectors. In 2012, the Higgs boson was found here. So that's pretty awesome. So that's why, you know, we're using this data and stuff, and we were furthering our knowledge about this, you know, the Higgs boson. So the Atlas detector is the primary detector I used. It's one of the two largest detectors at CERN. So the other being CMS. So the detectors are both set up for, both CMS and Atlas are set up for the most general range of physics. So it can detect the most general range of physics just because, you know, we want to detect as much physics as possible from these giant detectors. So there's four major components inside of the detector itself. It's the inner detector, which measures the momentum of each charged particle, the calorimeter, which measures the energy of the neutral and charged particles, the muon spectrometer, which measures the momenta of the muons, which is a type of particle, and the magnet system, which bends trajectories of charged particles so that we can measure their momentum as well. Cool. So the problem we're looking at, so there is several outstanding properties of the Higgs boson, right, that we want to measure. So these are things, we've basically been using the Higgs boson as a tool to discover new physics. So if we test something happens and, you know, a prediction of ours is different from then what happens in reality, that's pretty much an indication of new physics, and we need to go back and maybe revise our models or see if something went wrong there. So basically we have this di-Higgs production versus the top quark background production. So the problem with this is there's two different signals, right? And we're trying to measure the di-Higgs production, which is one of the outstanding properties of the Higgs boson that we are still trying to measure. The problem with this is that the top quark background, which is another process going on, happens much more often, and it's a lot bigger of a signal. So what we want to do is basically more accurately model the top quark background so that we can measure the di-Higgs signal, because they have the exact same two final states. So they result in the same thing. There's only very little difference in between the two. So the methods we used. So we used Monte Carlo simulations, which is basically you have a probability function that generates a set of values, which we then use the standard model to determine a physical situation. and then it goes back and the Monte Carlo runs another probability, and then we run it again. And we keep running it until everything outputs into these histograms that we have here. We can see this on the right. On the top plot is the histogram, is the actual variable that's outputted. So we can observe below it, this is called, we make a ratio of this plot. So this blue line here, you can see the blue and the red and the yellow, and they're all going like this. So the blue line is the most accurate that we've seen to the real world. And this is also a Monte Carlo simulation. So what we're trying to do is take the other Monte Carlo simulated variables and make a ratio of the two. So we have, on the bottom, we have this plot that we know is really accurate. And then we're putting the other data on top of it, and we're outputting this ratio essentially to the bottom here. So if they're exactly the same, they're going to be a 1-to-1, and you're going to see that these points here lie on the line, on the one line. But you can see over on the left side, they're kind of all over the place, which would indicate there it's a bit more uncertain. So one of the methods we can also do to try to fix this is to create new variables to be used on deep neural network training. This would be, so we take this variable we create, we put it into the Monte Carlo simulation, and then hopefully it makes it better for us in the long run. So some results and observations. Here is one of the uncertainty reducing methods that we attempted. So we have, you can see the transverse momentum, which is the little PT on the left side of LJET1, and then we have PT of LJET2 on the right side. Now you see we create a ratio of these two histograms, this top plot here. So we basically divide them by each other, and then this outputs another variable. This variable is what we can use then in deep neural network training to hopefully improve our measurements or our predictions, so that we can have a better prediction of this top quark background, so that we can accurately say when we detect this Dihiggs signal that, yes, this is the Dihiggs signal. So the conclusion is, so we see that modeling variables across different Monte Carlo generators differ. And one way we're trying to, one way we try to reduce these differences between the Monte Carlo generators is by creating these new variables and placing them into deep neural network training. We can also see from the data that we've searched through that the MCAT and LO variables are poorly modeled. So they need to be more accurately, they need to be worked on. Their simulations need to be worked on. And we can continue to minimize these uncertainties in the top quark background. So some acknowledgements. I'd like to thank Dr. Suyog Shrestha for being my mentor on this and my advisor. Dr. Derek Tex for also being my advisor during this. I'd like to thank Dr. George Kaiser, Dr. Ed Tolsky, and Dr. Carl Kem, all for recommending me this, as well as Dr. Tex for recommending me to get this internship and to work with this excellent opportunity with Dr. Suyog. And I'd like to thank the Toll Fellowship Program for allowing me to pursue this research. And that's it.
Great job, Peyton. I'll unmute myself so you can hear some clapping. Any questions for Peyton? You can either type them in the chat and I can help moderate, or you can just simply unmute yourself and ask your question.
Yeah, and if you guys want me to go back to a slide or anything, feel free to ask.
We have one question in the chat. What is a Dihiggs signal, or Dihiggs signal, excuse me?
So it's the Dihiggs production event. I'll move this back. So this right here. So the Dihiggs production is when it's basically two Higgs are basically produced, and then they they separate, and then you can see they have these final, when they kind of decay, they have these final different states. So that would be the Dihiggs signal. It's what we're detecting. It's the Dihiggs event, and we're measuring. That's basically the signal we're trying to detect. So it's the The signal is what we're trying to measure, and the Dye-Higgs event is what happens when the particles collide. And that's something we're trying to, and that's basically the main thing we're trying to measure the properties of. Because if there's a deviation in what we predict versus what actually happens, well, we see there may be a problem there with our theory. Or it confirms it, and everybody's happy.
All right, another question from the chat. How can it be predicted that two protons will collide at Atlas versus anywhere else in the 27-kilometer loop of the collider?
So I believe that's just from where they release them at, right? So they should you know, have the right trajectory and the right velocity in a certain direction, and then they should end up colliding there. This is what I believe happens.
I can add a sentence. It's more or less like that. The protons are going in clockwise and counterclockwise directions in two beam pipes, and they are brought to collide. Like the beam pipes are crossed inside the Atlas detector, and so that's where they collide. Well, and the CMS detectors, and then the others, but that's more or less it.
And I have a verbal question. So I've been paying attention to the Large Hadron Collider for a while. How did you get such a cool opportunity to work with data from it, right? It seems amazing that you were able to do that.
Well, thank you. Yeah, I was really thrilled when I had the opportunity. Really, I've been telling my professors for a really long time, and I have to give this to them and just the opportunity of Dr. Suyog, and kind of the stars aligned, right? So when COVID hit, you know, I was applying for research experiences, and all my professors knew that I wanted to do some research experience, and I wanted to have this research experience at an undergraduate institution, or, you know, just do some sort of research, because I had yet to do any up to that point. And then Dr. Suyog was actually is coming to be a faculty member here in the physics department. And he said he had an opportunity for a potential summer research student, and I was recommended by my professors. So really can't thank them enough. And yeah.
Sounds like a great thing to have on your resume that you've worked with that data. It was very cool.
Any last question? All right, really excellent work, Peyton. You guys are always so impressive. Just amazing.
Thanks,
okay.
Everybody.
Yeah, next we're going to transition to Shannon Thomas. So again, Shannon, introduce yourself, what department you're in, what major or what year you're in school, and who you work with.
Hi, everyone. I'm Shannon. I'm a senior environmental science major, and I've been working with Dr. Van Meter, who is part of the environmental department, but also the biology department. Okay, so as a Toll Fellow, I've been working with Dr. Van Meter on some salamander research. The first portion that I'll talk about is in the fall, we were able to go out and do some salamander field sampling. So here's just some photos of us with the salamanders. We explored one of the school's RAFC sites during the marbled salamander breeding season, which is around September and October. You basically just walk around and flip debris to look for the salamanders, so large rocks and twigs and tree trunks and things. And then once they were found, we would pinpoint their location on a GPS, take a photograph to measure them with image J, which I'll show in the next slide, and then swab them for pesticide exposure. So in the lab, here are the salamander photos that we would use. The swabbed Q-tip will be analyzed for pesticide uptake, but I haven't worked on that yet. The portion that I've been working on so far is salamander snout vent length, which is the picture here, measuring their snout vent length, and then the surface area. So the software ImageJ uses, like, we can tell right here that we have 1 inch, and so we can use that reference to tell the whole surface area of the salamander or their snout vent length. There was also an experiment that we did last April, which unfortunately, obviously I couldn't be on campus for, so I was just kind of over Zoom doing data collection. But we were testing salamanders' uptake of pesticides and how it impacts their glutathione levels. So the salamanders were exposed to soil that was sprayed with pesticide for 8 hours. It was either 2,4-D, which is an herbicide, or chlorpyrifos, which is an insecticide. And they were then swabbed to test their dermal uptake of the pesticides. And then later their brains and livers were extracted for analysis. And that is what I've been working on recently. So before school started, I was working for 2 weeks with Dr. Van Meter in the lab, prepping solutions and homogenizing the brains and livers so that they could be put into a 96 well plate pictured here. And then there's a plate reader at the college that can give us the measures to figure out glutathione levels from this. So we worked a lot, mainly with practicing how this procedure would go. And then we did some basic data analysis. So the plate reading spits out an absorbance value, and then these were plotted as a function of time up here. These are just our standards. And this is called the I slope. And then the I slope is plotted as a function against the concentration of our working standard, which is called the F slope. And then these numbers are used to calculate the total glutathione values for each sample. That's all. So we're still working on it. Actually, just today in the lab, we ran another plate and are starting to get some actual real numbers. So we've just mainly been working, all the data that's here is just like practice so that we really make sure we're doing it all right. And then after we get the numbers from the April experiment, the solid glutathione levels, then we'll be able to work more with the swabs that were taken from the field. So that's all from me.
I have a quick question.
Yeah.
What is, I'm going to butcher the pronunciation, but glutathione, what is that show in the lizards?
It's so, okay, I haven't worked with it very much, and I am like more on the environmental science part of it and not the biological side of it. So I'm not super familiar with it. It's just a natural like hormone in the body, in the amphibian's body. And so the theory is that when they're exposed to pesticide levels, it has an impact on their glutathione levels.
Thank you.
I have a question, Shannon.
Yeah.
I was wondering why you chose those two particular pesticides when you exposed the salamanders. I was also kind of curious what's on the swab when you're swabbing them. Do you know what chemicals are on it, or what, or is there anything on there?
I think the swabbing is to test for like the pesticides that they might have been exposed to, and then they'll like dermally, it's like on their skin, and then we would see like predict that if it's on their skin, then they're going to uptake it and it might have an impact on their glutathione levels. I cannot answer the pesticide question. I don't think I was part of that decision.
Oh, that's okay. I mean, they're pretty common pesticides. I was just kind of curious. Yeah, thank you. Very interesting.
I can answer quickly if Shannon would like for me
Yes.
to this. Okay, so the swabbing is part of a larger effort that Jen Wanat and a colleague over at Hartford Community College and I have started about a year ago, where we're going out and sampling salamanders at a bunch of different sites to figure out how land use surrounding the wetland impacts the biological functioning of the salamander, but we're trying to develop a non-lethal method. So like Shannon said, we go out and find the salamander, swab the skin, and then the swabs can be used to detect the presence of pesticides on the skin. But we're also hoping that from the swabs we'll be able to measure glutathione, acetylcholinesterase, and corticosterone. So those are all common biomarkers of stress response in amphibians after pollutant exposure. So that's why we're tackling those. And the reasons why we chose 2,4-D and chlorpyrifos are that, well, chlorpyrifos has been pretty controversial. People were hoping it was going to get banned because it has some really adverse side effects, but it wasn't banned. And so the EPA just re-registered that. So we wanted to collect more data on that. And 2,4-D is another commonly used herbicide that has, I mean, it's been around for decades and decades. I could talk all day about different pesticides. I won't, but it's just 2 common use pesticides that we know have adverse impacts, and we're trying to collect data to support amphibian risk assessments.
Great, thank you.
All right, we have another question in the chat. So what sort of implications does the change in glutathione levels have on the salamanders and the ecosystem? Does the amount of pesticide exposed to the salamander change the results? So if they have a small dose and a chronic exposure versus a single large dose.
Sorry, I'm just reading that over to make sure I
Yeah, it's a detailed question.
Yeah. Okay, what sort of implications does the change in the glutathione level?
Sorry, it was my question. I could just explain that verbally. Yeah, so okay, first part. So the glutathione levels changing in the salamanders, what does that actually do to the salamander's health? And then how would that translate in a response in the ecosystem?
So the glutathione levels are a measure of like the salamander's stress response to the pesticide. So I don't know if the glutathione levels could necessarily, I mean, I guess if they're exerting like high levels of glutathione, that would mean that they're very stressed out in their environment.
Okay, thank you. The second part was, I'm wondering about if a small, more repeated chronic dosage would change the amount of like stress response in the salamanders as compared to like a single large dose.
That is a very interesting question and certainly could be answered by a whole other experiment that is done. But that's a really awesome question because yeah, like when they're in the field, they're not just exposed to like pesticides all at once, they're like naturally exposed to it all the time. So yeah, that's super interesting, but I can't speak to that with the research here.
Well, thank you, and good, nice presentation.
Thank you.
All right, Shannon, there's another question in the chat just about the general role that salamanders play in their environment. So what do they do?
Okay, so I guess amphibians in general are thought to be like super important to their environment because they're like biphasic. They exist like in the water and then also on land. And they're typically like their skin is very sensitive to like the factors in their environment. So specifically something like this, where there's pesticides in the environment, amphibians are typically like very sensitive to them. So that's why Dr. Van Meter focuses so much on them because they are really telling to what like levels of pesticides or other pollutants might have on the rest of the environment.
Lots of great questions for all of our presenters. All right, nice job, Shannon. One last round of applause. And then our last presenter today is Martina. So typical drill, introduce yourself, your year, your major, and who you worked with.
All right, so hi everyone, my name is Martina Towers and I am a senior biology major and I'm currently working with Dr. Wanat. There we go. So we are currently looking at investigating the role of the yeast gene NPL3 in senescence. So to start off, what is senescence? Senescence is an irreversible cell cycle arrest. This is where the cells will remain viable and metabolically active, meaning they're alive and doing all of their normal functions. They'll just cease to continue dividing. And this is important in a lot of areas, such as development with embryogenesis, some age-related diseases such as osteoporosis and Parkinson's disease. And then also has roles in understanding cancer and developing potential cancer treatments. So senescence occurs in response to any event such as oxidative stress or UV damage that would put cells at risk for transformation through genetic alterations. And this is an alternative to apoptosis in that instead of the cell dying and losing all function, it maintains its functions but just stops dividing. This can also be induced when telomeres become critically short. And telomeres are these protective caps at the end of chromosomes that protect the genes near the end from being exposed and getting damaged. But what happens is that during DNA replication, these telomeres will get shorter and shorter until they become what's called critically short. And when they become critically short, those genes at the end are exposed and can become damaged and elicit a bunch of other responses. So senescence kind of serves as an anti-cancer mechanism, as when the telomeres get shorter and shorter, they would either senesce or apoptose. However, if they choose to bypass that and not senesce, they can create what's called genome instability, where they are either mutated or one of their favorite things to do is join chromosomes together when they're not supposed to, and this causes a lot of damage. The cell can also apoptose at this point. However, if they can figure out a way to re-lengthen their telomeres, this is how we get a lot of cancers. So telomeres are normally lengthened by the enzyme telomerase, and telomerase is quite a few proteins working together, but it's really this RNA template which will go down the line and lengthen a telomere to keep it from becoming critically short. However, this enzyme is only normally active in stem cells and germline cells in humans, so not in our somatic body cells. Telomerase reactivation is found in around 85% of human cancers though, some being melanomas and thyroid cancers, however, and a lot more. Cancers can also utilize what's called ALT, or alternative lengthening of telomeres, to maintain their telomere length. And this is a recombination-based mechanism where they will use other chromosomes and other telomeres as templates to lengthen their own telomeres. And this is found in a lot of sarcomas, brain tumors, and pancreatic cancers. We don't really know how the cancers can pick which mechanism they use, whether it's telomerase reactivation or ALT. It's not very well studied, but there is evidence that cancers that utilize ALT tend to have a poorer prognosis. So that is something that is currently still being researched. And to study this in the lab, yeast cells make for a really great model system. They have a lot of homologous or similar genes to humans. However, they have telomerase active all the time. So to overcome this, we delete what's called the TLC1 gene from their genome. And what this does is it removes their RNA template, so their telomeres will shorten with each cell cycle and then eventually experience that cell cycle arrest, senescence. And similar to ALT found in humans, they'll form what's called survivors, where they will find a different way to lengthen their telomeres and continue dividing. So another great thing about yeast is that they can be both diploids with 2 copies of their genome and haploids, so only 1 copy. And what we can do is we can take a diploid heterozygote, so 1 copy of TLC1, and sporulate it, and we will get 4 daughter cells, 2 of which having that TLC1 gene, so their telomeres are activated, and 2 without. And since they all came from the same mother cell, we know that they've inherited telomeres of roughly the same length, and that they didn't start dividing until, or the telomeres didn't start getting any shorter until this point because up until then, they'd have this TLC1 and their telomerase would be active. So what we can do is then run what's called a senescence assay on the haploids that have the genotype that we're looking for. And a senescence assay looks like this, where we are looking at the cell density on the y-axis versus the population doublings on the x. And population doublings you can think of kind of like time. And what we're doing is we are counting and diluting cells every 22 hours and really looking at how much they're replicating. So when they're not senescent, they're replicating a lot, so the cell density is a lot higher. And when they stop replicating a lot, that's when we get this point of senescence. And then after that, the survivors take over, and since they can elongate their telomeres, they will replicate a lot faster and take over. So looking specifically at our gene of interest, NPL3, it is an RNA binding protein, which has a lot of interactions in the cell. It does a lot of things, some of note being transcriptional elongation, so helping make RNA from DNA. It also helps in translation, so making protein from those mRNAs, and then also helps maintain those telomeres. And looking at a unpublished senescence assay that's already been done, we can see that the wild types and the NPL3 mutants that still have their telomerase active don't senesce. They're nice straight lines. However, when we remove TLC1 and they experience that cell cycle arrest, they senesce around 75 population doublings. And then if we further remove NPL3 and TLC1, we see that the senescence is around 68 population doublings, so it's faster. So what we're looking to figure out is why does deleting NPL3 result in this accelerated senescence? So to help figure that out, we're also looking at what's called TERA. TERA is a non-coding RNA made from telomeres that basically signals that the telomeres are getting shorter. It's also been shown to increase during senescence, and it's basically telling the cell to senesce. So again, what we're looking at is, does NPL3 and its transcription properties influence the transcription of TERA, or is it working with somebody else to influence this transcription? So to look at some of its potential interactions, we are looking at what's called the PAF1 complex, and that's this guy over here. It has a lot of roles in transcription, from elongation, termination, to associating with RNA polymerase II, and then also controlling how open the DNA is to the transcription machinery. So looking a little closer at the complex, we are looking at CTR9 and PAF1, which on top of all their roles in transcription, have also been shown to affect telomere length. So what we're looking at is whether these proteins could potentially interact with NPL3 and influence the transcription of TERA and thus senescence. So to do so, I designed overexpression plasmids, so circular pieces of DNA, and we put our genes of interest in there under the GAL1 promoter. And under the GAL1 promoter, in the presence of galactose, whatever gene follows it will be upregulated, so it will be overexpressed. And we're looking at, will this overexpression compensate for the accelerated senescence that we saw in the NPL3 TLC1 double mutants? If either of these proteins can compensate for that accelerated senescence, it would indicate that they're functioning within the same pathway and that they're interacting. So looking at the senescence assays, we can take our diploid heterozygotes and sporulate them with our expression plasmids. And then we can run the senescence assay on the haploid progeny expressing, or with the TLC1 NPL3 double mutant, and then also with the plasmid. And from what we saw before, TLC1 senesces at a certain point. We'll run the assay in glucose, so that way our expression plasmids aren't making a whole bunch of the extra protein. And it would look really similar to the TLC1 NPL3 double mutant, which has that sped up senescence. And then we would also run the assay in galactose, which would overexpress our proteins of interest. And we would see if that would what's called rescue the phenotype, whether it would make up for that accelerated senescence, and that would indicate that they're working together, whether it has no effect on it, or whether it would further accelerate that senescence. And by looking at these results, we will be able to tell the interactions between NPL3, PAF1, CTR9, and how it influences senescence. Any questions?
Great job, Martina. Again, you can type your questions in the chat or you can unmute and just ask away. All right, questions in the chat coming in. You mentioned something about deleting the TLC1 gene from the yeast cells. How do you do that?
Yes. So, I actually don't think I have a good slide for that. So, to delete the gene, there's already I believe a set library of deletions that have already happened. So the strains were already made by the time that I got to them. And then what you can do is just mate them with different ones that already have the same or have the mutations that you want, and then you can select for the genotype that you're looking for.
All right, another question for you. Do humans have TLC1 or NPL3?
So humans do not have TLC1. Humans have this. The human telomerase complex looks quite different, but what the TLC1 is most similar to is this, what's called H-TERT, which is right here. And that is what houses that RNA template that's going to be used to extend the telomeres. So it's similar, but not the exact same thing. And then NPL3, it has what's called it's homologous to it in humans. It's a heterogeneous, like ribonuclear protein, where it has kind of the same functions as it does in yeast. It just has a different name.
From a presentation standpoint, we had a question about your diagrams and how did you make or get them?
Which diagrams?
Evan, do you want to unmute and ask?
Yeah, you had some really great visualizations, like the one on that previous cell and the ones with that are like representations of the actual like layers of things going on.
Oh, well, some of them are modified slides that I got from Dr. Wanat, such as this one, and these are figures from the paper that we're working on. So they're kind of just from her, and I've modified them and put some extra animations on them, but thank you.
Effective uses of PowerPoint animations, right? It's great. Any other questions for Martina, our last presenter of the evening? All right, well, let's have one last big round of applause, either digital emoji hands or literal hand waving. You all did a wonderful job. I'd welcome everyone, especially our Toll Fellows and Apprentices, to hang out afterwards with our presenters and ask questions about what it's like to do research with faculty. Thank you, everybody.